That's a cheeky question, and you're going to hold me to that, aren't
Of
you?
course. Why do you think? Why do you think we're recording this?
Absolutely.
This is the Convergent Science Network podcast. Leading researchers in the domain of neuroscience, brain theory, and technology are interviewed by Paul Verschure and Tony Prescott.
It's Paul Verschure with the Convergent Science Network podcast, and I'm here with my colleague Tony Prescott. We're both in our BCBT summer school here in Barcelona 2015, and I'm here with Narender Ramnani, who was giving us actually a very different kind of view on cerebellum, because the standard view on cerebellum is really like, well, this is sort of this little appendix that hangs off your brainstem, and that might be involved in some form of motor control. But when things really get interesting, cognition, decision making, forget it. We go look at cortical areas, mainly prefrontal cortex, and you sort of turn that whole perspective upside down. So how did you come to that perspective?
Well, that's an interesting question. So this goes back again to everything being, the foundation for my views are really the anatomy of the system. And so one of the things that I became very curious about was the looped architecture of the cortico-cerebellar system, and this idea that our knowledge of what the cerebellum is talking to is incomplete. And then over the years, the gaps started to become filled, and some very nice work came out from Peter Strick's lab and from the work that Schmarman and Pandya did shortly before that. And this work really highlighted the idea that the cerebellum isn't simply talking to the motor cortex. There are many other parts of the cerebellum that are communicating with really quite a diverse range of different areas of the neocortex, such as the prefrontal cortex, and in fact many different parts of the frontal lobe, and also the parietal cortex. So there's this diverse range of inputs. So the question is, if it's just a motor structure, what on earth is it doing? Just doing motor control. Mm-hmm. And so that's really where it all kicked off from.
Right. You began also by talking about what is now quite a classical view of cerebellum, sort of the Maurer-Albus theory. Could you just summarize for us again? And also, do you think that that's still a useful framework to think about cerebellum?
Oh, undoubtedly. I mean, it's a framework that hasn't changed very much, actually. I mean, people have tinkered around the edges with it, but the fundamental message of what Maurer and Albus were talking about is still very much there. And the perspective that they offer is, the theoretical perspective that they offer is this idea that Purkinje cells are the computational unit, the principal computational unit, and that this is a system that's capable of plasticity for supporting learning, and that learning is achieved by adjusting the inputs into Purkinje cells, and these inputs are typically parallel fibers, and those changes occur under the guidance of a teaching-related error signal from the inferior olive. Now, having said that, there are new perspectives emerging. They also talk about adjusting the synapses between cerebellar units and their inputs. And I think it's recognized now that this is not the only source of plasticity in the cerebellum, but you also have plasticity involving a lot of other cell types in the cerebellar cortex.
But one of the basic messages out of that is that there's a sort of microcircuit, and that it's repeated across the cerebellum. Do we still hold with that?
There's very little data to argue against that view, and so the microcircuit or the microcomplex idea is still something that's very much in play, and the anatomy and physiology work bears that out, really, particularly in relation to the work that's been done using eye movements or using classical Eichmann conditioning, where people have identified very specific microzones that might contain the Purkinje cells that house that plasticity.
So, but as you were saying, sort of classically, people have thought this microcircuit is doing something in the motor domain.
Yeah.
But you now want to generalize it.
Absolutely. So the architecture of the cerebellum is considered to be quite uniform. It's really very, very different from the architecture that you find in the cerebral cortex, where cytoarchitecture is quite diverse. So people will have come across the concept of Brodmann areas in the cerebral cortex, where the area 4 has a different profile of cells from its neighbors in area 6 or wherever. The idea about the cerebellar cortex is that it's much, much more uniform than that. So the computations that are going on in any given location will be the same in one location as in any other. So that idea still holds up, although people are starting to find some interesting biochemical differences that suggest that there may be some interesting but subtle differences between these areas. So one of them, for example, is the idea of there's this substance called aldolase C, or zebrin, that's differentially expressed in different parts of the cerebellar cortex. And one of the interesting things about zebrin is that if you stain the cerebellum for zebrin, you get a stripy parasagittal organization. That in itself tells you something about the parasagittal functional organization of the cerebellum, but it also suggests that maybe there are some subtle differences in the way that these adjacent zones compute as well.
Right. Mm-hmm. And how many zones do we think we have, say in the primate?
Oh, crikey. That's a difficult question to answer, because I think as you go out more laterally, they become more and more refined. And the other thing, of course, is there are slightly different ways of
measuring it,
of characterizing a zone. I mean, zebrin is one way, but there are many
because I think
subzones. I think
the system I know better, the basal ganglia, has similar notion of a microcircuit which is repeated across.
Yeah.
But when you drill down and ask people to define the microcircuits, it all gets very hazy.
Yeah.
And you end up, well, some people say there's larger domains, of which there might be a handful. And some people will say, well, there's very small microdomains. I mean, so is there still a controversy in the cerebellum about the granularity of the domain?
I think there probably is. I mean, there's still this slightly old-fashioned controversy that continues to rumble on about whether you have patchy somatotopy that's determined by the parallel fiber inputs, or whether there's this other organization that's determined by, zebrin. But the other interesting thing I should say about zebrin is that it seems to correspond very closely with one of the important input systems from the inferior olive. So climbing fibers project to the cerebellar cortex almost on a one-to-one basis. So one climbing fiber will completely dominate the physiology of any given Purkinje cell, and it seems as though the parasagittal organization that we find with zebrin seems to correspond with the parasagittal organization as determined by the projections from the inferior olive. So there's an interesting there's an interesting story there.
So the microcircuit is really defined by the climbing fiber. And
arguably, that's what's been claimed by some authors. Yes,
absolutely. So you also made the claim that there's sort of this co-evolution of cerebellum and cortex, and that in that sense the cerebellum performs like a universal transformation from its inputs that it receives over the pons to its outputs that go out over the deep cerebellar nuclei. But also, as Maren Albus tell us, this is all dependent, how this output is shaped is all dependent on the error signal you get from your inferior olive. So Now, in the case of classical conditioning, it's fairly easy to understand where that error signal comes from because it's linked into the periphery that just says, okay, you just got shocked on your orbit or you got an air puff on your cornea. But if we now say, well, a huge chunk of cerebellum is actually bidirectionally interfaced to prefrontal, what's the error signal in that case?
Well, that's another really fascinating question. I think one of the problems that we're confronted with is that there's so little data, so little physiological data that can answer that question. And it kind of highlights how much work there is left to do. But some of those questions could be answered in terms of the anatomy. We know, for example, that there are parts of the dopamine system that encode prediction error. We also know about the fact that elements of the dopamine system are sending their outputs to the cerebellum. And so there's a route there for this information to reach it. So, for example, the VTA not only sends its error signals off to, quite large areas of the prefrontal cortex, but interestingly, there are collaterals that branch off from those projections that end up forming connections with Purkinje cells, and these are dopamine connections. So it's as if the cerebellum is receiving a copy of the error signals that are reaching the neocortex.
Okay, so you're saying maybe for the interaction with frontal areas, inferior olive becomes less important to convey error, and it will be more derived from a VTA-dependent dopamine signal, which might signal something like a reward prediction error. That would be the idea?
Well, that's one thing that one could claim. I think we shouldn't underplay the importance of the olive. There is recent data that has demonstrated there are in rats prefrontal inputs to parts of the olive that then project on to areas like CRUS1 and CRUS2 that receive input from the prefrontal cortex, from the other system. So there's a lot of different anatomical routes through which that could happen. And one of these areas in the prefrontal is the anterior cingulate. And we know that the anterior cingulate does play a major role in the monitoring of errors that are cognitive in origin, errors in your own cognitive processing, for example. And those areas do seem to send in rats an output to the olive, and then the olive then sends those projections back to...
For you, which area would that happen? Would that then play out in rat? What areas are we talking about?
Which neocortical areas? So the anterior cingulate cortex. I don't remember off the top of my head which particular anterior ACC areas, but Tom Rickrock has basically characterized that system.
And it would then just run over the thalamus to the inferior olive, or this is a collateral.
No, this would go directly from the neocortex to the olive, and from there on into cerebellum. There are direct inputs,
and these are topologically mapped. That means these anterior cingulate projections that end up in the inferior olive target climbing fibers.
Yes,
that target regions in the cerebellum that are again recurrently coupled to that same bit of neocortex.
That's right, because the study was conducted using transsynaptic traces, and because these traces have the ability to jump synapses, it's possible to do that point-to-point connectivity.
Right, so that's cool, right? Because that means you're heuristic to say, okay, in case we don't understand, let's follow the anatomy.
Yeah,
is then again paying off. At least the idea of having the inferior olive as your error signal generator,
yes,
is then still intact also for these.
Absolutely, and I think the other thing it does is to open up the possibility of physiological studies to try and investigate these circuits at a functional level.
Right, but now to finish up the inferior olive.
Okay,
I just want to say that. The other thing, which however I felt I didn't see back in your diagram in a too prominent role, is a negative feedback between the deep nucleus and the inferior olive. I mean, it might sound like a detail, but if it turns out that these Purkinje cells are actually actively controlling the errors that they receive over the inferior olive. So is that kind of error feedback control that occurs within the cerebellum for you computationally less relevant at this stage, or just something that you've ignored for other reasons?
I think it's an extremely important facet of the circuit, and in fact, I think, if I'm not mistaken, there was an inhibitory output coming out in those diagrams that I showed, projecting back to the olive. And one of the things that this seems to be involved in is the damping down of error-related activity. So, for example, in classical eye blink conditioning, you'll be aware, perhaps more than I would, that this pathway is important for decreasing, in some ways, the salience of the unconditioned stimulus. Jerry Heslow's work has shown that and Chris Yeo's work has also shown that the amplitude of the US is decreased in the presence of a conditioned response. So if there's an efference copy, if you like, of the CR that leaves the cerebellum, and on its way, it basically goes down back to the olive and reduces the salience of the US. And there are some interesting parallels that one could apply that to in the cognitive domain. If there's a system like the cerebellum that's specialized for feed-forward behavior that doesn't really benefit from feedback, and indeed could be impaired by any sort of ongoing feedback if it causes an interruption to the processing, you do want a system that dampens down the effects of any ongoing error feedback coming into the system.
So I wanted to clarify about your diagram. So you have the inferior olive feeding back to 3 areas of cerebellum, and then you have VTA feeding into another area. Is that right?
Yeah.
So is VTA is dopamine signaling something similar then to what the inferior olive is signaling in these other areas?
Well, the diagram that you're talking about is simplified, and there are a few speculative things about it. But in essence, there will be some things that are similar,
right?
But there'll probably be some things that are different too. So, in answer to your question, so what would be similar? One of the things that it could signal are the higher level cognitive errors that we spoke about earlier. What could be different? Again, this is speculative because nobody's ever sat down and recorded from both of them to see how they differ. But the dopamine system signals reward-related error, and so that's why I've included it in that pathway in that diagram.
Yeah, I mean, so you have these overall domains of cortex, which are, I guess, they're defined in part by where they get their input from in cortex.
Yeah,
but then also they're defined in part by where they get their feedback from. Yeah.
Yes, although I'm not ruling out the possibility that the olive could also be feeding into the high level circuits that I was
right
that I mentioned.
Okay.
I'm not ruling that out. It's just that we need to see some sort of concrete evidence for their existence before we could do that.
Sure.
So then, but now, so you presented us with a really fascinating diagram, which I liked a lot, and there's a lot to discuss about it. But of course, there's also data behind it, and I think what's really impressive is that you've been able actually to replicate a lot of the phenomena that we know about cerebellar learning in humans. So could you, to what extent is, let's say, at least in your mind, is the response of the human cerebellum actually identical or similar to what you might expect in a rodent or a macaque under classical conditioning conditions or VOR? So
Yeah, so one of the things that I alluded to in my talk was this idea of a conditioned stimulus being processed similarly, regardless of whether it's involved in the process of classical conditioning or whether it's involved in the process of instrumental learning. Mm-hmm. And so one of the things that I found very interesting was to basically draw from the classical conditioning literature, because things are so very well known in that area. And one of the things that I mentioned was the idea that when you record from the eye blink microzones in the cerebellar cortex, you find a decrease in the activity of the Purkinje cell in response to a conditioned stimulus during classical conditioning. So the issue is that if there is such a thing as a universal cerebellar transform, where a similar form of information processing is applied to, let's say, inputs going into a part of the cerebellar cortex that's receiving from prefrontal cortex, if you take a higher level form of learning, for example, like instrumental learning of rules, the question would be: do you see something similar? Do you see a decrease in the activity of the of a patch of cerebellar cortex when a CS is presented at the same time? And the work that I mentioned seems to bear this out. There seems to be a decrease in activity.
Which is important, right? Because on the one hand, you replicate what we would expect from physiology, which is we obtain a pause in Purkinje cell firing, would mean less oxygen consumption and reduced blood flow. But it also means the pause is not dependent on another, let's say, population of interneurons that now inhibits the Purkinje cells, because then your blood flow pattern might not have changed so much. Would you agree with that?
Absolutely. And I think one of the things we have to be very careful about when we interpret imaging is, what does an increase mean? Because this could mean an increase in inhibitory activity, which could be manifest in a different way if you're using electrophysiology. One of the things that was most, very interesting about the work that I've just mentioned that uses electrophysiology is the use of gabazine. And so gabazine inhibits the action of inhibitory neurotransmitter inputs onto Purkinje cells. But the CS-related decrease that you observe is still there, despite the fact that the gabazine has acted on the system. And what that suggests is that the input that's driving this inhibition are probably the excitatory inputs coming in via parallel fibers. Mm-hmm. And the value of understanding that for imaging is that if you have a decrease in activity during imaging, it's probably the case that there's a similar explanation involved that doesn't involve the ramping up of activity by increased metabolic demand, mm-hmm. for example, from inhibitory to neurons.
Yes.
Is there a sort of subtleties about how you interpret the fMRI data with cerebellum? Because when you have learning, you actually have decreased activity. Is that right, generally? So as you see learning proceed, you don't necessarily expect once the system is tuned up to see a lot of activity there.
I think sometimes it's very difficult to interpret this sort of data. We know what we can say is that it's consistent with the electrophysiology, but there are studies that show learning-related increases in the cerebellum too. And of course, we will never know why, because we don't know what cell types are generating that information. We don't know we basically don't know what is driving that activity. So, for example, in a study of learning where you see an increase, that could be that you're ramping up activity in the cerebellum for other reasons. It may not be that you see this sort of Mar and Albus type change that I spoke of before, or this albus type decrease that we were comparing with. It's possible that you're getting inputs in that are ramping up the signal from other areas of cortex for other reasons. Learning-related activity is not always going to be manifested as a decrease in the cerebellum.
So what's the strategy then when you have this?
It's a hard one. Yeah. Imaging is a great tool to use for many reasons. I mean, not least because you can image the entire brain in one go, and plus the fact that you could look at anatomically very specific areas and so on. There are lots of benefits to imaging, including the fact that you could do it in humans. You can examine tasks in humans that you wouldn't otherwise be able to do in animals. But the key thing to remember is that fMRI is otherwise a very imperfect tool. Every tool has its drawbacks. And with fMRI, you're not going to be able to tease apart things like which cell type is contributing to my signal, and the indirect nature of it, the fact that you're depending on a secondary measure of electrical activity rather than the electrical activity itself. So there are lots of things that get in the way of the interpretation, but it's still an excellent tool to use.
Right.
But now, so the paradigm you've used on humans, which is really interesting, sort of moves away a bit from the traditional division we have between operant learning, going back to Thorndike, and what's called classical conditioning, going back to Pavlov, where in the first case, we are learning about the effect of our own actions on the world. And in the second case, the world exposes itself to us, and we have to pick up whatever correlations are in that data. So you have tried to find a paradigm where you really merge instrumental aspects of learning with classical aspects. So how do you exactly balance that? How do you make sure that you can really still call it instrumental?
Well, we can call it instrumental because of the nature of the contingencies in the trial design. So with instrumental learning, well, let me start with classical conditioning, where, classical conditioning, you have a conditioned stimulus, and that will always be followed, regardless of what the subject does, by an unconditioned stimulus, which will cause an unconditioned response. And so there's no contingency between the way that the subject behaves and the outcome. The outcome will always be delivered. So that's fine. That's the classical that's the classical scenario. The instrumental scenario, of course, is very different. We make sure that the outcomes are dependent on the subject's behavior. So there's a very clear contingency there. So the trial design really takes care of that, I think.
Mm-hmm. But now, in the operant case, you would also believe that, let's say it's something like there's a larger time interval we're talking about, and also the feedback the subject gets has a different kind of quality. So in this case, your human subject, I guess, is not being shocked anywhere. So we're not talking about an aversive stimulus that's being applied. So do you think that is a qualitatively change for the cerebellar learning system, or actually it turns out to be still operating in an identical way, in your mind at least?
Well, that's an interesting question. I think that the best way to answer that is just to make clear that the inputs that come into the cerebellum come in through, so it's basically in the trials that I'm using, in the methods that I'm using, I use a visual feedback. There are lots of reports showing that the cerebellum has access to that information, at least at a sensory level. So whether, and classical learning theory will tell you that the more intense a stimulus, the faster you learn, and that sort of thing. Those same kinds of manipulations can be applied to instrumental learning. And so you can have a partial reinforcement schedule and so on. the kinds of signal that we see coming from the cerebellum seem to mimic the amplitude, well, the rate of learning, I would say.
Mm-hmm.
And so they all seem to follow these sort of Rescorla-Wagner type rules in that sense.
But now when the task becomes instrumental, we also have to start to think about how do we represent a rule that's hidden in this task, right? And how do we then pick up the relationship between that rule and my own actions? So in your mind, it is that rule representation that would be the frontal neocortical contribution, and the action generation would then be the cerebellar contribution. That's how you think about it?
Yeah. So the not really. I mean, I tend to think so what you're suggesting is that the rule representation is neocortical, and that the action related representation is coming from the cerebellum. I don't follow that path at all, because if you again look at the wiring diagram, it doesn't support it. So
I find it surprising, because I thought we just had agreed that all the wires are there to link these two systems together.
They are, but if you look carefully at the system, the cerebellar modules are all separate from each other. They don't talk to each other. The only scope for the cerebellar signal to influence the neocortical system, or rather, the only way in which these modules can communicate with each other is through their links via the neocortical areas that do wire up to each other. So you've got prefrontal, premotor, primary motor cortex, all talking to each other a lot. The cerebellar modules that are independently wired up to these areas are operating independently of each other. There is no wiring within the cerebellum that can allow them to talk to each other.
So you're saying they have to operate through the neocortex?
Yeah, basically that's right. Yeah.
But in theory, they could also, let's say, via brainstem motor nuclei like the red nucleus, the pedunculopontine nucleus, you might loop back to the pons and then generate as an efference copy and input to a next cerebellar circuit and loop it straight through the cerebellum. Would that be anatomically defendable?
Run that one past me again. I missed that.
So an alternative is that I send my motor command from the deep nucleus of the cerebellum down to the red nucleus, or to any other brainstem motor nucleus, pedunculopontine nucleus, from which I get an efference copy back into my pontine nucleus, input station to the cerebellum, and now I can loop it over a next cerebellar circuit. So I think in theory, anatomically, you could devise a scheme that they can loop multiple cerebellar circuits without ever talking to the neocortex. Would you buy that?
I would probably find that. I think if the pathways existed, then I would, I think that may be supportable. But what is the evidence that the anatomy exists that could support those? That could support that.
Well, red nucleus for sure projects back to inferior olive, for instance.
Yes, it does. But I think the olive is a very modular structure, and so the red nucleus is not going to project to every part of the olive. Similarly, does the red nucleus, to what extent do different elements of the red nucleus communicate with, eventually with areas like prefrontal and premotor cortex?
Well, I can play it another way to see if since you like the wires, I could also say, okay, we got an efference copy, execute the motion, comes back to somatosensory cortex, and from there straight into your pons. Right. So still, I would be able to loop multiple cerebellar circuits without ever talking to my motor cortex.
I think the cerebellum is a very, very modular structure, not just intrinsically, but also the way that it is wired up with lots of other areas. I think all of the evidence to date suggests that these loops don't communicate with each other. And it's a generalization in some ways, but I think, we have to go with that because that's the evidence that we have available.
But is it important to your model, at least how you think about the circuit functionally? Is this an important axiom of your model? Like that if it would be violated, the whole model collapses?
Well, that's an interesting question. There's no computational instantiation of my model yet. So there hasn't been an opportunity to sit down and write this into a piece of software and test it.
But it makes it much easier to speculate about it. That's a good thing.
Yes.
Your model sort of claims the system 1, system 2 distinction.
Yeah.
And the system 1, which is where you say the cerebellum has the lead role, is a sort of habit learning system. But what you described about the anatomy would seem to suggest that the habits have to be enacted via the cortex, even if they're learned in the cerebellum.
Yeah, because I think the apparatus for delivering the habit-based behavior is housed in the neocortex. It's housed
it could also be housed in the brainstem, elsewhere. There's a lot of machinery there for controlling the motor system, where you would not need motor cortical input.
That's right.
And you could imagine that, with the cerebellum, you could wire up all sorts of interesting responses in the brainstem without having to bother with motor cortex.
I think that's a possibility, but the neocortical architecture that supports very complex behaviors is better placed, I think, to execute those kinds of behaviors than the circuits in the brainstem.
So in that case, system 1 would be about wiring up circuits in motor cortex to perform the habit behaviors. So is it more of I'm not sure how this can work if the loops don't interact. I guess you've got your loops in motor cortex, but what's my prefrontal cortex loop? How's that able to help me learn a motor habit? Or am I learning some other
No,
kind of
you're
habit?
learning these modules are learning independently.
So what's habit learning in the context of prefrontal cortex?
Okay, so what habit learning in the context of prefrontal cortex is, for example, the execution of a rule, the execution of, for example, the kind of experiments that I talked about earlier, where you're arbitrarily pairing, a stimulus and a response in a completely arbitrary manner. Now, you can think of that as having motor control demands. You can think of that as having sensory demands, but you can also think of that as having cognitive demands. It's the cognitive element of that is being basically being run by the prefrontal cerebellar loop.
Yeah, but to me, this doesn't make any sense, really. I'm sorry to say, but look. The point is, so let's start with Schifrin and Schneider, right? So we talk about controlled versus automatic processing.
Yeah.
And it also means the controlled, deliberate system figures out the rules. It figures out how to map sensory states to action states given goals. With that, you train your habits that now can be executed by your automatic system. And why do you want to do this? Because you want to be fast. You want to optimize your response latency, right?
Yeah.
Or you want to free up resource in frontal cortex.
Yes, absolutely.
Well, I would think a real fitness requirement is latency more than working memory space, but we can debate that, okay? So here I am, now I have my automatic process. What am I optimizing? Time, okay, latency. So now, also in the perspective of Kahneman, you would have system 1, system 2. System 2 is the slow system, it's a controlled system, deliberate, and then system 1 is a fast automatic system. Great, okay? But now you're proposing that this fast system that puts in this huge amount of metabolic resource into getting fast, and the Purkinje cells and the cerebellar circuit is fast, right? And it really controls latency very precisely by disinhibiting the deep nuclear cells. So you rely on the rebound, which is controlled in latency to trigger an output. But that's not what you're building on. You say, no, these guys are really maybe very precise. But again, that signal goes back to motor cortex, which sits there, a piece of cortex, it's integrating, it's sloppy, it's jittery, whatever. And then that piece of cortex is going to issue this final motor command that cruises down my cortical spinal tract to control the skeletal muscle system. But I'm adding latency now. Why would I do that?
Because you have to parcellate up different elements of the task. So, I mean, what you're suggesting is that somehow this very high-level thing that you've learned finds its way into the motor system without going through the motor cortex. But there's no route. I mean, how would it do it? I mean, if you think about the connections of Crus 1 and Crus 2, which are the parts of the cerebellum that are supposed to be learning these things, they have no output to the motor system. So there is no other route.
Mm-hmm. But maybe they're doing something else and controlling action. Maybe they're part of the slow system, and it helps the slow system to figure out, let's say, interval information, timing between events. And it is not involved at all in generating actions. For that, you train up other parts of the cerebellum.
Yes, well, that's basically what I'm suggesting. So you have modular parts of the cerebellum. So you have some parts that are specialized for dealing with prefrontal problems, some parts dealing with the primary motor cortex, and it's basically speeding up elements of the information processing that would otherwise go on in each of these neocortical areas. But you need the neocortical areas to actually deliver the response. There is no output from Crus 1 and Crus 2 that can access this corticospinal tract, except through going back to places like prefrontal cortex and then, sending the signal down through premotor cortex and primary motor cortex.
You also exclude that there's any kind of divergence in, let's say, the deep cerebellar nuclei that might allow them to indirectly target brainstem motor nuclei.
Well, that's an interesting question. So the perspective that Peter Stricker's offered us is that these are highly modular systems. Now, the missing information in all of this anatomical work that we've talked about before is the absence of any point-to-point connectivity that's been shown. So it's never been shown, for example, that there's a particular neuron in the primary motor cortex that sends its outputs to specific Purkinje cells, and that those specific Purkinje cells send outputs back to thalamus and back to that particular neuron in the primary motor cortex. So that point-to-point connectivity has been missing from the literature.
At the single cell level.
At the single cell level.
But it does reach that volume of cells.
Yeah, so the broad areas are, so it's been shown, for example, that if you put anterograde and retrograde tracer into M1, you'll end up with label in the same cerebellar lobules. And that suggests, quite broadly, but it suggests that there are these independent loops and that they don't communicate with each other. However, I think to be conclusive about it and to be sensible about developing computational models, you have to demonstrate that this point-to-point connectivity exists.
Well, I'm not sure if that's a fair constraint, really, because certainly, so what is the shortest distance at which people have found these recurrent projections to terminate? I mean, we're talking about, hundreds of microns, or this will be the scale, right? So as soon as you hit cortex, you have a highly excitable substrate, which dense local coupling with lateral coupling. So as long as you target, let's say, an area that is, let's say 100 micron away, you can be quite sure that activity will percolate down to that neuron.
Well, that's right, except that the things that we were talking about earlier were about ways in which inputs from prefrontal cortex can start to influence
the motor output,
right, and so on. These are in different lobules altogether.
Sure, but I wasn't. Indeed, we weren't finished with that, and I have a solution. Because maybe the thing to consider is that even though there's a universal cerebellum transformation, the cerebellar areas that are interfaced to prefrontal don't care about action generation in terms of the motor plant itself, but they assist prefrontal areas to figure out, let's say, interval information about task relevant or decision variables. Because that's the unique contribution they can give you. They can tell you something about interval timing. And this is as much necessary by acquiring the habit as it is for executing the habit. So how about that?
That's plausible, I guess.
So not all is lost for my canoe right now.
It sounds like a compromise to me. It sounds like.
Oh, really? I'm already watering it down. Now. Okay. So good. So now so we have different interpretations of how, let's say, the dense interaction of cerebellum with prefrontal. Would you say, how much of the hardware, the cerebellar hardware, is committed to interacting with these premotor areas, you would say?
The premotor areas?
Well, or prefrontal, just decision-making areas in the brain, let's say.
Estimates have varied, but a very, very significant chunk of it. I mean, I think Peter Strick, in one of his papers, if memory serves me correctly, he said that there was something like 40% of the territory of the cerebellum in primates was accounted for by the motor system.
Then including the decision-making parts of what we now call motor system.
No.
So
the issue is, what is the rest of it doing?
Yep, okay.
And in theory, very significant chunks of it are going to be devoted to it, communication with prefrontal cortex. And it sort of makes sense that that's the case, because prefrontal cortex in the human brain has expanded enormously during the course of human evolution. And if you take on board the fact that connected systems tend to evolve together because of the selection pressures being applied on systems as a whole, then yes, that sort of makes sense. It makes sense.
But I find that such a strange heuristic, because I might also imagine I'm an engineer, right? We're the God engineer, we're building brains. We have these modules, as you say, right? It has a very modular structure, the cerebellum. So I think also Tony mentioned that in the talk. You could imagine I engineer some animal who has lots of really fast movements in a complicated dynamical world. I give it a huge cerebellum, and it doesn't need to solve any problems. It doesn't solve crossword puzzles, and Wisconsin card sorting is also not available in the jungle. So it just reduces the neocortex. So why do these things by necessity have to co-evolve?
Well, by necessity, I mean,
that's what you seem to say, no?
I'm suggesting that if selection pressures apply to systems that operate functionally as a unit, then you're going to find an expansion on the whole. You're going to find an expansion of the parts of the cerebellum that are communicating with the prefrontal cortex. And that's indeed what we've found.
And this expansion at the Purkinje cell level, the granule cell, all of them together?
Those are empirical questions. We have no idea.
But one thing you did say was that there's a kind of a dorsal ventral, I think, distinction in the amount that PFC talks to cerebellum. So there's much more connectivity with the dorsal area.
Well, there's a dorsal ventral split in the dentate. So right. the dentate nucleus is you can split it up in terms of the dorsal part, which is wired up with the motor system, and the ventral part, which is wired up with the prefrontal system.
Right.
And it's been shown that previous studies have shown, when you compare across primates, that there has been a selective expansion of the ventral part of the dentate nucleus. And we're now starting to explore those sorts of issues. I
think I was referring to, so elsewhere in your talk, you were saying that there were parts of prefrontal cortex which have a strong projection, other parts which have a weak projection.
Exactly, yes. So one of the interesting things that was found by Jeremy Schmarman and Deepak Pandya was that the dorsal parts of prefrontal cortex, so areas that are dorsal to sulcus principalis, have a greater tendency to project to cerebellum than ventral parts.
Right.
And so you could speculate about why that might be. And, that might tell us something about the kinds of information that the cerebellum wants to process, as it were.
But now the other, let's say we agree, okay? So it all evolved together, and
very pleased
then we can talk about basal ganglia, hippocampus, and spiracolliculus, see if they also co-evolved the same way, and they might have done that or not, okay?
Yeah.
But so now we have a control system which definitely will depend on neocortex, frontal areas, and basal ganglia. I don't know, maybe you want to leave that out, I don't know. But now I have to compile my habit into my cerebral circuit, right? So how do I first segment a habit? And then secondly, how do I get those segments into my cerebellum that I can call them up on future occasions?
That's a really interesting question and a tough one to answer.
We need answers.
Yeah, absolutely. Absolutely. So the issue of how do you segment a habit, I think that's a tough one. I think all you can say is that if there are associations being formed, if there are CSs being processed in areas of the prefrontal cortex, then there'll be a spike train corresponding to that going down to the cerebellum via the pons. How you can sort of break that very high level representation down and talk about it in terms of what will the spike train look like and how will that be represented in terms of Purkinje cell activity or whatever. I think it's not possible to answer that. Mm-hmm. It's a tough one.
Do you think it will exploit the segmentation? Do you think it will exploit already the structuring of action primitives or behavioral primitives at the brainstem level? Because as a cerebellum, if you take the eye blink conditioning analog, I am talking to my red nucleus. I'm triggering discrete behavioral primitives.
I don't think so.
Okay.
And the reason for that is, let's think, for example, about the kinds of information that gets processed in the hierarchically organized neocortical system. So if you take, for example, cells in the prefrontal cortex and you try to figure out what they're most interested in, the work of Earl Miller shows that they're interested in rules. They're not interested in the fundamentals of motor control. You won't find motor unit activity being represented in the cells of the prefrontal. Equally, if you look at cells in the motor cortex, you won't find them being interested and having their response properties tuned to particular rules as you would find in the cells of prefrontal cortex. So I don't think that you're going to be... I don't think it's a useful exercise, really, to try and look in places like the brainstem for things that are, rule-related, as it were.
But still, if we think about a simple rule, which is like later on you might want to escape from the studio and go to the lounge, something like this, right? And if we do sufficient or enough interviews with you, then you would be having this habit of running to the lounge and breaking all the furniture. Okay, so that's a habit now. But this habit will have primitive elements, which might also be getting up out of your chair, controlling posture, going to the door, moving through a door. So they're very simple elements in the end that get stringed together. So what is special about the habit is its macroscopic structure that even brings you from here to the lounge where you smash up all the furniture. So the point is that with these primitive elements, we can easily account for at the brainstem level, no? Like just to get up, maintain your posture, locomote.
Can you? I'm not so sure.
Okay.
So give me an example. I mean, the kinds of representations that you have at the very lowest end of the motor system, would be things like the control of individual muscle units and so on.
No, look, but if you go to the midline structures in your brainstem, where you have the central gray, for instance, where you have whole behavioral patterns that are species specific, encoded in a local way.
There'd be sort of hardwired things down there.
Sure, like, and there might be gaze controls, completely predefined reticular formation,
so there'll be sort of aggression and all of that sort of...
Sure, but they're behaviors, right? They're behavioral programs that relate to certain motivational systems.
Yeah.
So you don't see them as part of then the habit. You really see the habit as targeting, let's say, higher level behavioral elements that you might have learned.
I think it's possible. It's perfectly possible for those lower level behaviors. So if you think about Ma's idea, I'm building everything on Ma, as it were, and what he claimed was that these higher level representations can trigger the pre-learned representations in the cerebellum and cause them to execute a particular kind of behavior. There's no reason, I think, why maybe the brainstem could participate in that. So, I mean, many years ago, the work of Leighton and Supple showed that there were cerebellar inputs. If you lesion the cerebellar vermis, you get sham rage. It's possible that that's happening because of the impact that cerebellum has on those lower level brainstem centers.
Right. Okay. So you're saying it's not necessarily in the focus of your idea about how habits are learned or expressed, but you cannot exclude it.
No, that's right.
Right. Okay.
Yeah, absolutely. I mean, there's lots of evidence showing that there's interactions between brainstem centers and the cerebellum, of course.
So the other thing that you mentioned, or that also becomes apparent, as you said earlier, in some sense you're saying, look, all this, the modeling of the cerebellum is like a footnote to Mar and Albus.
Yeah.
Right? So you really think that there has been not that much progress in our theoretical understanding or computational understanding of the cerebellum beyond what Mar and Albus have identified?
I think there's certainly been some progress, of course. You can't deny that. We've come on leaps and bounds since 1969. What I would say is that the fundamental ideas about cerebellar plasticity and the way that they work have not changed all that much. The idea that the cerebellum has as its most fundamental unit the Purkinje cell, and that the inputs into the Purkinje cell, the parallel fiber and the climbing fiber inputs being, the principal inputs into the Purkinje cell, is still a dominant idea. That's not to say, of course, that we shouldn't be exploring other forms of plasticity in the cerebellar cortex. There are lots of other cell types, and it's very clear that there's experimental evidence to support their involvement as well.
So here we are, right? So you have this really very, if you want, ambitious and also advanced scheme where you see really how prefrontal cortex works together with cerebellum to realize both, let's say, controlled processing and automated processing, right? So, and in that sense, I guess you also have had to sort of fight your battles and, accumulate your scars to get that point across. So, given your experience now in trying to understand the brain at a system level, what is Narenda's law that we should follow to understand the brain at this level?
There's no law as such, but
of course, come on.
I think we just have to be faithful to the data. I think the starting point, if the starting point is the anatomy, then I think you're on very sound footing. It needs to be based on that sort of evidence. And the other thing is to build from what we have learnt in the motor system. The motor system is what we understand best. So if we can take a set of principles that we can apply in the motor system and find a way of extending those into the cognitive domain. I think that would be a good way forward.
So look, Tony likes traveling, and then he always has me pay it, so I don't like to spend money. So we only have trips to within England in the coming 5 years. So 5 years from now, is going to come to your lab.
Absolutely.
To check whether a prediction you're going to make today was actually confirmed or rejected. So what's the most important hypothesis that you want to see tested, validated in this 5-year framework perspective?
That's a cheeky question, and you're going to hold me to that, aren't you?
Of course. What do you think? Why do you think we're recording this?
Absolutely. There are so many interesting questions. I suppose one of them would be to go on testing the idea that a conditioned stimulus is processed in the same way in conditions of instrumental learning and classical conditioning. We've often talked about the possibility that there is this universal cerebellar transform. And it's an idea that people have been on about for a long time and for good reason. I think this is a good way of doing that. I think it's a good way of pinning down that problem and saying, well, look, you've got a conditioned stimulus. Is it being processed in the same way, regardless of whether it's higher level or lower level? Are the areas that are activated when you process that instrumental CS, how are they talking to the cerebellar cortex? Or indeed, are they speaking, to the prefrontal cortex? Which parts of the prefrontal cortex are they talking to? We still don't have a handle on exactly what areas of prefrontal cortex are saying to the cerebellum. And so I think it would be good to try and work out methods for sorting out the communication between the two.
Great. Narender Ramnani, thank you very much for this conversation.
Thank you.
Thank you.
Pleasure.
Well, that was fun.
You gave me certainly a good grilling there.
The CSN podcast was produced by the Convergent Science Network of Biomimetics and Biohybrid Systems.
I enjoyed it, actually.
Project funded by the European 7th Research Framework Programme.
I hope you still consider it friendly. But
of
For
course.
interviews, recorded lectures, or upcoming conferences in the field of biomimetics and biohybrid systems, go to csnnetwork.eu. And thank you for listening.
I think this is the idea of the podcast, that we can go a bit behind what you would put on the slides, right?
Yeah,
and also, I think it's important for people who listen to this to understand, look, there's uncertainty in what we do.
Yeah,
right?
absolutely.
We make assumptions, and to get that a little bit more to the foreground is really, I think, what we want to achieve here. And if you want to also show a bit more, the more human level decision making that we have to do when you do these things.
That's often missing from talks, isn't it?
Yeah, exactly.
But no, it's really good fun. I enjoyed that.
Mm-hmm.
Terrific.
I enjoyed it. No, actually, we were really, I'm also very much trying to figure out this whole idea of how do we compile habits, So indeed, how can the cerebellum contribute to that is quite relevant for what we are doing. So that's why I at least
it's a question of trying to understand what is a motor habit and what is a cognitive habit, really, and trying to
right, exactly.
And that's something that, I think I could have answered that a little more effectively. But it's a tough one. But
you have to admit, right? It seems really odd to claim it loops back over M1.
Yeah,
you're screwed. You add lots of uncontrollable latency to this whole thing.
Yeah. There, a big chunk of the timing is taken up with the decision related aspects, and that's what you speed up with that prefrontal component.
But maybe we give too much importance to M1. I think M1 is a pretty stupid structure.
But there's no way out of the system without going through M1.
Yeah, but I see M1.
How can you
really
reach the corticospinal tract?
No,
without
wait,
getting
exactly.
M1?
But you just dump your motor program in M1. You give it a very short time window to play it down into the corticospinal tract, and you close it up again. That's it. Finish. It's just the output station,
Yes, but how do you get it in?
Via your premotor network.
Premotor network being through red nucleus and so on.
Wait. In my model, my cerebellum does not need to talk to M1. It doesn't give a damn about M1. It just plays it out downstream immediately because it wants to be quick.
Yeah.
I think also from an evolutionary perspective, that's the one thing that matters. This is what makes you survive. Be quick.
Yes.
That's why you want to do this at all.
Yes, I understand.
Right?
Yeah.
So for that, this is why you just want to play it straight down.
I suppose the crux of the issue is how do you get information out of Cruise 1 and Cruise 2 and into your premotor network. And I don't think there is a route except through, what would it be?
Well, look, Cruise 1, Cruise 2, they are talking to deep nucleus. You might have divergence.
Yeah.
We cannot exclude that. This is the thing we have to check.
Actually, right.
And
okay.
I would bet, certainly since it's brainstem, damn sure you have divergence.
Yes.
Right? So
then you throw away modularity, right?
Yeah, but I wasn't.
Well, maybe that's okay.
I wasn't committed to modularity. That was your problem.
Right. Yes.
But we still have conserved it at the cerebellum level. But outside of the cerebellum, I just allow things to diverge or to be linked together. You still, for me, just the key is why don't you allow lateral interaction cerebellum? Because you get latency. You say, if I go through that loop, I want to get out in a predictable way. There can be no screwing around.
This is why it's important to figure out the intrinsic connectivities within the cerebellar nuclei then.
Absolutely.
And the data's not there.
Are you sure?
In primates, it's not there.
I should ask Björn Merker. He might know. He's a good friend, great expert on brainstem. We should look at red nucleus, for instance. I remember discussing this with him a few weeks ago, but not so specifically for the red nucleus. He didn't reject the idea completely, but I didn't ask for the references. I'll ask him.
Yeah, I a few years ago, I think this issue did bother me, and I looked. I couldn't find the anatomical evidence.
Okay.
And the simplest route out was through M1. Possibly because we know about the connections.
Sure.
And we don't know about the connections, the intrinsic connectivity within the deep nuclei to allow that convergence.
I think I did well because I have two options now. I have plan B, right? Plan B is the prefrontal oriented cerebellar regions don't care about linking to the motor output. They just care about setting the right interval information for even the amplitude time course of, let's say, a memory response prefrontal.
Yes. Yeah,
so they
absolutely.
don't give a damn about motor.
No.
Right?
I think that's more likely, actually, because, the language is different. The language that the prefrontal cortex speaks, when I say language, I mean,
the neural activity.
The neural activity and what it's time locked to and so on, it doesn't give a damn about motor. Exactly. It just doesn't.
Exactly right.
And so maybe it isn't bothered. Yeah, I would buy that.
Because then what it could look like is that you just have this prefrontal cerebellar interaction. Cerebellum helps keep time in that system. You get your deliberate action plans you play out over M1, goes down over the cortical spinal tract, but you have your collaterals into a range of brainstem areas. You have your efferents copy back to the pons. You have a lot of source information for the cerebellum now to suck in these regularities. And then from then on, I would speculate, right? It would just play this out automatically, exploiting recurrent projections into the pons. I just allow to loop multiple circuits.
Yeah.
Now, the function of this will make sense to me. Then I really have optimized my latencies. This is what I want.
Yeah. Okay? Yeah. I think there's a lot of merit to that, actually. Yeah, I agree.
Just the anatomy is missing, but that's a detail.
a forgettable detail. I suppose.
Exactly. But I'm sure, given that people haven't looked, it's a reasonable hypothesis to go to check. So I should convince some rat runner to do some tracer studies.
Yeah, or even somebody who works on monkeys. Yeah, even better.
Or humans. Why can't we do this? Isn't there a way to do this? Can't you look at brainstem areas?
Paul, I'm going to have to go and make a hotel.
All right, so then we see you at 8:30 at Silken for dinner?
Yeah.
Possibly?
Yeah, okay, I'll try to come.